Good evening, and welcome to the Open Minds Teen Science Cafe. I'd like to thank all of you for coming out tonight and greet our online audience tuning in. So, if you have a question online, you can put it in the comment section and we will answer it during the Q&A. So, please remember to be respectful to all other guests and silence your cell phones if you haven't already. And we will be taking pictures during the cafe to post on social media, so check us out on Twitter, Facebook, and Instagram at Teen Science Cafe RAL. So, tonight we have not one, but two guests, and both of them are PhD students at NC State, one of which likes backpacking and the other ultimate frisbee. So, please give a warm welcome to Erica and Nelsita.
Hello. I'm glad you all are excited to learn about butterflies tonight. My name is Erica Henry. I'm a PhD student at NC State, and we're going to talk about some of my research today, but I also do research on rare butterflies and habitat restoration down in the Florida Keys, where I'm headed tomorrow.
Hi, everyone. My name is Elsita Kikabush, and I'm also a PhD student at NC State, and I also study butterflies. And my research focuses on climate change and how that affects butterfly life cycles. And we're really excited to talk to you today about a very special butterfly.
Yeah, so we're going to give a little talk about the research that we do, and along with other folks in our lab, and then we'll do answer some questions, and then get to the activity after that. All right? So we have a bunch of slides that will be on these monitors. Talking about our work to rescue, restore habitat for rare and endangered butterflies. But when I say endangered species, I imagine a lot of you think of these. Pandas, rhinos, species that are big and exciting and charismatic on other continents. And then when I say, no, I study an endangered butterfly, people are like, oh yeah, I know what you're talking about, maybe. Should I point this somewhere else? Oh, there it is. Does anybody know what this is? Monarch, yeah. Everybody's heard of monarch butterflies, right? And while monarchs are declining, these are data from monarch counts from the mid-90s to today, and you can see that those bars get shorter as you go down the graph. There are still millions of monarch butterflies in the world, perhaps even billions of them. And so we do not study monarchs, and we are not going to tell you about monarchs today. Instead, we're going to talk about other butterflies and Lepidoptera that are also in decline. This is a graph from a research study that looked at all Lepidoptera, which are the butterflies and moths in the globe, and looked at what their trends have been in the last 40 years. And you can see the yellow line on that screen shows that they're all declining. So this is not unique to monarchs, and it's not unique to the species that we study. It's a global issue. That's exciting. So what we're going to talk about today is the St. Francis Satyr, this pretty little brown butterfly that you can see on that leaf there. St. Francis Satyrs are about this big, maybe like 1.5 inches. And they, I got ahead of myself. Does anybody have any ideas on what causes endangered species declines? Yeah. Habitat loss. It's like you've seen my slides. Habitat loss is by far the biggest driver of species declines across the globe. I have a picture here of the longleaf pine habitat in the southeastern United States. And that light gray on the map is where longleaf pine used to be prior to European settlement. And the dark gray is where the longleaf pine forests are now. And you can see there's a tiny bit of it that still comes up into North Carolina. And that's where St. Francis Satyrs are. And we're going to talk about that today. So this is a butterfly that is super rare, and it only occurs in North Carolina. And it only occurs in North Carolina where that red X is on that map. And that red X on the map is Fort Bragg, the Army base that's just down by Fayetteville. And we've outlined the Fort Bragg in yellow on that map, and you can kind of see, or I notice when I look at the map, that there are big places where there's intact forest. And when you looked outside of the yellow line on that map, it's all neighborhoods and farms and cities, and it's all developed, and there's tiny little bits of natural areas in there, but not the big tracts of open natural habitats that exist on base. Maybe. And Elsita's going to tell you more about the butterfly.
Okay, so the St. Francis Satyr, I'm going to talk to you a little bit about its life cycle and its biology. So kind of a refresher. So butterflies go from eggs to chrysalises, from eggs to caterpillars to chrysalis to adult butterflies. And they start as caterpillars during the winter. If you look on the left of the graph, they sort of hibernate, or they go into diapause over winter where they're not developing, they're just caterpillars. And then when the temperatures start increasing during the spring, they start developing and start eating the plants that are growing, and they start developing, and then they pupate into the chrysalis in late April. And then they emerge as butterflies in May, and the adults kind of all fly around, and they mate, and they lay eggs, and those eggs hatch, develop into caterpillars, pupate, and they have another flight period of adults flying around, mating in July. Those lay eggs, the eggs hatch, and those diapause again through the winter. So the pretty important part about all this, the connection to habitat, has to do with their host plant. So the host plant is the plant that the caterpillars eat and they survive upon in order to grow to become butterflies. And in the case of the St. Francis Satyr, that plant is a group called sedges. They're sort of like grasses. They're in the wetlands. And they kind of exist in these big open habitats that look like this, kind of nice and open. Now, there's a couple factors that cause disturbances that cause this perfect open habitat that you can see here. So one of them is fire. Now, fire was native historically, pre-European settlement, and it was caused by Native Americans lighting fires and also by lightning. But now, sort of in the modern day, we control where fire occurs a lot more, and in developed areas and cities, we don't really want fire to be in our backyards. So a lot of the fire that sort of naturally caused fire-tolerant species like sedges to proliferate is now not occurring. So without fire, looks kind of like this. Lots of shrub overgrowth. But at Fort Bragg, active management, sort of lighting fires and doing that in a prescribed way, this causes those open habitats to occur. And additionally, artillery fire also causes fires to occur in the impact areas. So military training actually causes a perfect habitat for species like St. Francis satyrs. So this is actually a picture of the open habitat in an artillery impact area. And this happens to be where the St. Francis satyr occurs, and also where other endangered species occur, including the red-cockaded woodpecker, the rough-leafed loosestrife, and the Venus flytrap.
All right. So, in addition to fires, beavers are also really good at maintaining open, beautiful, sedgy wetlands. Beavers also have been removed from the landscape over the last century or so. They were extinct in North Carolina for a while. They were hunted to extinction because people like their pelts. Now people move beavers around when they build dams in places that they don't want them. And so what we do in our lab is we try to be the beaver. And we try to recreate these open sedgy wetlands outside of the artillery impact areas. We don't really work in there much. And we cut down trees and we build dams. You can see in this picture what we use is an aqua dam, which is just like a big plastic tube that we use a pump to fill with water from the stream. And then the weight of that water sits on the ground and acts like a beaver dam. So you get a pond-ish kind of on one side. We cut down all the trees. And when we do that, now it's going too fast. How come you can do this and I can't do this? When we do this, we get nice results. We get nice open sedge-dominated wetlands. So you can see the before totally shrubby and awful wetland on the left and the nice open sedgy meadow on the right. But we do this often in places where there are no butterflies to begin with. And so once we have habitat that we think looks good for St. Francis satyrs, we go into those artillery impact areas and we go there twice a year because this is the place where there are the biggest populations of St. Francis satyrs. So we go in there, the middle guy in that picture is actually an explosive ordnance division person that we have to take with us to make sure that we don't step on anything that hasn't exploded. And we catch butterflies, and it's awesome. Sometimes those EOD guys that come with us get super into it, and they're like, I was chasing a butterfly once when we were there that day, and I lost it, and the EOD guy was behind me, and he was like, it's over there! And I was like, you need a net. You're good. Anyway, so they come into the artillery impact areas to kind of like chaperone us biologists around. We catch female butterflies. We bring them back to a greenhouse that we have on base, and then we put the females in what we call oviposition chambers, where we hope that they lay eggs. Then we leave them there for 2 days. We let the females go back in the wild, and then we keep those eggs in the greenhouse where we put them in the enclosures that you can see on the right, and we allow their caterpillars to feed and develop in the greenhouse. And when they emerge as adults, we put them in the car. We put them in the car, we drive them to the wetlands, maybe. Yeah, to our restored places. And we release them. Eventually. Yeah, yeah. And sometimes this is like 40 minutes. So we'll just wait 40 minutes for the, and we release them. One thing that I forgot to mention earlier is that when the adults emerge in the greenhouse, we try to put them together in little net castles, which is what Ben, our technician, has in that picture, and hope that they mate so that then females can lay eggs in the greenhouse before we even release them into restoration sites. But our best success has happened during that 40-minute car ride where they haven't mated for 2 days in the greenhouse, but when we arrive at the restoration sites, they're mating in the car. And so then we have to drive back to the greenhouse. Oops, wrong way. I haven't moved it. And then every once in a while, working in the sand hills in North Carolina, there are very giant puddles, and one year one of our technicians totaled the car in one of them. And we haven't had any successful captive mating attempts since. So we're looking for new strategies, new field vehicles, etc.
So we've released the butterflies in our restoration sites, but how do we know that we've had successful populations propagating in those sites? So what we do is we count butterflies doing daily surveys. And so this picture is an overhead view of one of the plots that we've restored. And we lay boards kind of in this tic-tac-toe fashion or shape, and we walk that exact pattern every day, and we look for butterflies, and we count them. Now, that sounds pretty simple, right? But it actually looks like this. It's really muddy. There's lots of insects. There's lots of cottonmouths. The boards are kind of in and out of the water. You can slip and fall down pretty easily. And then on top of that, you're looking for tiny little brown butterflies that are flying around. Well, in short, we've found that restoration works. So this is a graph showing population sizes over time at one of our plots that we've restored. So this site, prior to us arriving there, was actually where the butterfly was first discovered in 1983. And at the time, the population was estimated to be around 300. But then, up until 1990, that habitat degraded, and that population, just at that location, went extinct. And so the butterfly was thought to be extinct for a little while until they were found on artillery impact areas. Around 2010, 2011, our lab started restoring that habitat by doing all those actions that we mentioned before, making fake beaver dams, cutting down wood, and also then reintroducing butterflies. And then doing our surveys, we've been able to estimate that our population has increased up towards almost 800 butterflies now in 2015, and we're still been counting onwards. I think it's you next. It's still me? Oh yeah, it's still me. Okay, so to kind of put this a little bit more into perspective, this is another butterfly. It's probably one of the most famous endangered butterflies besides the monarch. It's called the Fender's Blue, and this is kind of a similar situation. It's another butterfly that needs fire and needs a disturbance to maintain the habitat that it relies upon and the host plant that it relies upon. So this is kind of like another similar population graph in which they started doing restoration actions, they started doing prescribed burns in prairie ecosystems for the Fender's Blue, and the population increased. So this is a map of the United States, and all of those dots represent butterflies that are listed as threatened or endangered. And the large dots represent butterflies that need disturbances, such as fire, beavers, hurricanes, in order for their habitat to be maintained. So in summary, there's a lot of butterflies, and there's a lot of butterfly biologists that are out there trying to understand populations and understand habitat disturbances to maintain those populations and to carry out their conservation efforts. So thanks a lot. That's our little presentation. We'd like to take questions now about anything, really.
Have you done anything related to the area of Weymouth Woods?
So it's actually right near Weymouth Woods. Like some of our sites are right near there. So Weymouth Woods is the state park sort of directly to the west of Fort Bragg. So we haven't worked there. I've been there. It's really awesome. And I do know that they also do prescribed fire management there, because I think there are red-cockaded woodpeckers there. You know that? And I think there might be some other endangered species. So some of those management actions that are done there would be similar to ones that we do, but we haven't worked there specifically.
There are tricky things about reintroducing endangered species in new places. And so we've been working really hard at Fort Bragg to show that we can do this and it can be successful and it's worthwhile to think about doing it somewhere else. And so that's kind of where we are now, is thinking about how and where and when to expand off of Fort Bragg.
I don't know if you'll have the answers to this, but I noticed on the chart you had one that was in the Rocky Mountain West, one of the red dots. Do you know what, is that in the mountains or is it in the prairie area?
That one is it's the Uncompadre fritillary, and it's a high alpine species. And it's mostly threatened by climate change, where it's getting pushed to the tops of mountaintops and nowhere else to go.
Beyond habitat loss, are there other factors contributing to like the endangerment of these species?
Definitely. So climate change was mentioned, but also pesticide use is another one. What are some of the other?
A lot of it, I mean, overwhelmingly, it's habitat loss. But in different systems, there are things like pesticides are a big issue with monarchs because people spray herbicides and you lose a lot of milkweed that way, which is their host plant. Monarchs also have some interesting parasite dynamics. They have these little parasites that live in their guts, and they have been implicated in some of the decline, but like really if you had a lot of habitat, all those other things wouldn't matter as much.
So what do you think the population will be at in 2020, or what do you hope it'll be at in 2020?
That's a good question. Well, we were just showing you one site. So kind of one of our goals as conservationists and trying to would be to unlist the species from the Endangered Species Act. And in order to do that, there's actually a couple of specific criteria. So we would need to introduce it into, I think, 2 reintroduction sites, and also then prove that that population that we've introduced is viable in the future. And that viability is often estimated at like a 50-year future time point. So it's not just a question of how big will the population be, but it's also will it be able to continue to be and continue to propagate that way over a longer period of time, and then how many populations would you be able to add on? So I guess those are our goals, would be to fulfill those criteria.
With these disturbance-dependent species, a lot of times, like if we stopped doing restoration now, all the shrubs would grow up and the butterflies would go extinct again. So a lot of how many there are in 2020 depends on how much effort there is in continuing that restoration work.
Are the reintroduction sites located close enough to the population sites that they can meander between them, or are they isolated?
That's a good question. So some of them yes and some of them no, I guess. So the artillery impact areas are not connected, so they are able to move along riparian corridors, so using the river and wetland system to move along that, but they're on different rivers. So the ones in the impact areas aren't connected to the ones that are outside. There's a historic population that we're actually trying to get it to move along to try to occupy more area and increase. So we've basically restored habitat alongside where that original population was, or where it was centered. And so we're working on that, and that's kind of a work in progress. But we do also then try to sort of track where they are, and especially when we release them from the greenhouse, we've marked them. So if you saw in that picture with the mating butterflies, they have numbers on them. So we can actually tell, based on where we release them and where we see them next, how far they can travel, and we can try to estimate how far they're actually able to move based on that.
What is the role of the St. Francis Satyr in its environment?
Tricky question. So that's a really good question. It doesn't really pollinate particularly much, so many butterflies go to flowers and nectar, but this one hasn't really been observed by us to do that, maybe only a couple times on one plant. So it's hard for us to say that it has like a strong role. It certainly gets eaten by other creatures, by other predators, so it's like food for others.
I've seen, dragonflies get them. Those big orb weaver spiders, I've seen them stuck in spider webs. The caterpillars are herbivores, so they're eating plants. A lot of times when we think about like what does a species do, a lot of the impact that it has on a system has to do with like how many there are, or how big of an impact it can have. Like one panda can impact a lot more than one St. Francis Satyr, right? And so when you have these populations that are now like all of the butterflies, fit in my hands, there's only so much impact they can have, and it's hard to predict what kind of impact they would have on the system and role they would play if they were everywhere.
So Erica, I remember during our earlier meeting, you talked about some of the work you were doing in Florida with the hurricanes that came over recently. So could you talk a little bit about that?
Yeah, so I do similar work on those dots that are at the end of Florida. And I work on a butterfly called the Bartram scrub hairstreak and the Miami blue. And the Bartram scrub hairstreak occupies a similar fire, pine, rockland system. So I do a lot of work where we're doing the same kinds of things, where we're trying to introduce prescribed fire, and in places where we can't do that, we actually use a giant skid steer to get rid of all of the overgrown understory. And all of my research was just in the eye wall of Hurricane Irma like a month ago, and I'm going down there tomorrow to see what it all looks like. But we're really interested to see if maybe the hurricane acts like a fire or these other disturbances to kind of get rid of extra shrubs and palms and things that are growing underneath the pine trees. Hopefully that's the case, and the case isn't that 3 feet of salt water killed everything, but we'll see.
Just one last question. With the butterflies, with all these that you have on the map, are any of them migrating at all, or is they pretty much all stationary, and that's why they're endangered in that way?
I think a lot of them are stationary.
Yeah, I don't know of any of those listed species as being very migratory. Really, like, monarchs are migratory, and then there are some, like, populations of painted ladies that move around. But most butterflies are pretty stationary in one spot all year.
So I know you've talked about it already, the challenges that are presented when you're trying to count a handful of species in a very large area. Do you have any interesting stories about a time where you were out counting and something happened or something ate a butterfly, or or any experiences that you've had like that?
When you released a butterfly?
I released a butterfly. I released a butterfly once out of one of those net castles, and it flew its little dainty flight, and then it started going like this. Zip, zip, zip. And I was like, what? And it had been eaten by a dragonfly. So it had like 4 seconds of freedom. And I was like, oh my God, sorry.
Yeah, I don't know. I would say just with field work, you know, it's always an adventure. I mean, we see plenty of venomous snakes, and there's lots of mosquitoes and chiggers. And since we work with endangered insects, we can't wear any kinds of insecticidal things that would be bug spray. That's the word I was looking for. We can't really wear that since we don't want it to get on the butterflies, so we just kind of get eaten by the mosquitoes.
It's also interesting working on a military base with active training going on, where sometimes I've been at our release site, and the soldiers, we have all this tape around the outside, and there's signs that say endangered plant area. But soldiers still, and it says no military training, but soldiers still will like walk right through our restoration sites. And I was at the truck once, and I could hear them in the site. And when I turned around, these 4 dudes in their camo all went like this. And they like squatted behind the bushes. And I was like, I can see you in there. And they're always like, oh, I guess we got lost.
Yeah, we have to chase them out sometimes.
Okay, so Francis Satyrs, since they're like so rare, I guess, how were they first found?
So that's a good question. So they were actually first discovered by a soldier who was an amateur butterfly enthusiast while he was doing training in that area. And so that was in the 80s. But then sadly, it was listed, and then later he was actually found transporting them out to sell them. So he was actually busted a little bit later, the same guy who discovered them.
What are the Francis Satyrs like named for? Like what person?
Saint Francis. I don't know, sorry.
Does anyone have any other questions?
All right.
Should we go to our
Yes.
activities
Just
slides? Is it this?
Yeah, it is.
Nope. Uh-oh. 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 Killed it. I'm sorry.
So what we're gonna do? So when we put up that graph and we're like, the population is 800 butterflies, we don't like teeter our way down the boards and count 800 butterflies. We have to have methods where we count what we can see on a given day, but then we can estimate how many butterflies we didn't see, how many butterflies maybe emerged on the days between when we surveyed. So there's all of these like fairly, well, some more sophisticated than others, statistical methods that we use to convert the number of butterflies we count into how big the population actually is. And so we were going to do a little activity with you.
Go up. We want to build a video. Right? Okay, so just to kind of give you an idea of how hard it is to see this butterfly, I took a video of one that I saw in the field. So hopefully this will play automatically. And see if you can see the butterfly and follow it along.
In the middle.
It's right there.
It starts in the
right,
middle.
It's right in front of you.
It is. It's brown. It's going to fly. There it goes.
You see it now? You see it, right? It's really easy to see. Come on, guys.
Yeah, so you can't see them all, and they're really easy to miss because they're small and brown and you're balancing on boards.
And that's one that moved. So when it moved, it was a little bit easier to see, but if it was just sitting there, could have just walked right past it.
So what we're going to have you all do, you've probably noticed that there are some butterflies around the room, is you are at your table going to observe butterflies, count them, and then be able to estimate how good you are at detecting them. And we're going to use a method called double observer methods because you have two observers. And how this works in the field is that Elsita and I might stand next to each other, and we would independently count how many butterflies we see, and then at the end of our count, we'd be like comparing our numbers, and then comparing which butterflies we saw. And if there are always butterflies that Elsita would see that I wouldn't see, ones that I see that she doesn't see, and then butterflies that we both see. And we can use all of that information to estimate how many were there that we missed entirely when we counted them. Does that make sense? So that's the, you are gonna, at your table, conduct your own sort of point count for butterflies on your own, and then, yes. And then after your count, you can compare with one of your neighbors and then calculate your personal detection probability and the population size that you would estimate. And so you'll notice that there are, well, I'm going to show you an obvious one. There are big yellow ones, there are little yellow ones, and there are big white ones, and there are little white ones. So those are our 4 species. Will you go to the next slide? Yeah, see? It's something about standing over there.
All right.
So on your table, you have a data sheet that looks like this, and that you can orient however you want, whatever is helpful. You might want to like draw features in the room on there, and then independently, without your partner, I want you to tally and locate as many butterflies of the 4 species that you can see. And then once you're done with that, you can compare with somebody else sitting at your table and figure out how many butterflies you saw in common, how many you saw that the other person didn't see, and how many they saw that you didn't see. And there's a place at the bottom for you to tally species, and then the numbers you saw, your partner saw, you both saw. And then you can do the math at the end. And we'll put the equations up on the slide there. All right, does anybody have any questions? Yeah. With the top half, the question is, what do you do with the top half? The top half is so that you can write down where you see butterflies in the room, and that'll help you compare with your neighbor which butterflies you both saw. And then you calculate everything with the bottom half, the tallies.
So you do your counts in the bottom half. So first you draw where you see them, and then you count them and put that in your tally by species.
And then after you do this, we'll talk a little bit about detection probability and things, and we'll also reveal what the population size is in this room of all 4 butterfly species. Okay? And we can walk around and help you if you have questions.
So if you have questions, just raise your hand and one of us will come and help you.
Should just walk around.
I got it. You can leave it on. Cool. Just gonna actually put it up here. Perfect. And remember, it's like being on an airplane. The closest butterfly may be in front or behind you. Emergency exit. Come on. Come on. That was good. Nah, people have been on planes before.
How'd that go? Is everyone wrapping up a little bit? Finding your last butterfly?
Okay, I think we're gonna we just have a few minutes left, so we'll walk through
The math.
the math in case you haven't gotten there, and then we'll reveal how many butterflies are in the room. So I realized that on the data sheet it was a little bit confusing, and both butterflies are it's not the total that you both saw, it's the actual individual butterflies that you both saw. So there might be butterflies that your partner didn't see that you saw, but you want to keep track of which individuals you both saw. So that's the idea of, like, in the field, you'd write it down on the map and then compare your maps and be like, oh, these two are the same butterfly. These are different individuals. They use this method a lot for birds, more so than butterflies, but I use it in my research for butterflies in Florida. So if you did compare observations, you take that number of butterflies that both of you detected and divide that by the number that you saw, and that's how good you are at detecting a butterfly. So that number is like, given a butterfly is flying around, there's a 30% chance that you see it, or a 70% chance that you see it, or whatever that number is. I will let you know that our estimates for detection probabilities for St. Francis Satyr is that we see 30% of the butterflies. So our detection probability that we've estimated for the crews that do surveys is 0.3. So that means that we miss 70% of the butterflies that are flying around when we do our counts. Oh yeah, did people actually calculate their detection probabilities?
What did you guys get? Anybody want to volunteer?
Are you better than we are? In this nice static room?
Did you see them all? 100%? 95? Anybody want to volunteer what their detection probability was? Yeah, just shout it out.
What's that? For which species? The big white, small white ones? Nice.
Awesome.
Nice.
Great. Anyone else?
A 100% on the large
Nice.
yellow.
Of them all. Yeah.
Cool. And then, do people have any population size estimates for any of the 4 species that they would like to share? 25 of each. 25 is close for one of them, but they're not the same number of all 4 species in this room.
I have a question.
We got the big white butterflies being 21, the small whites being 21, the big yellows being 17, and the small yellows being 18.
Nice.
Interesting. What about you?
The little white ones had 25.
25. All right,
Nice.
so you ready for these totals?
Yeah.
Big yellow, 23. I feel like we're playing bingo or something. Little yellow, 43. Big white, 33. Little white, 46. What?
Anybody see in the 40s for some of those?
Was anybody close on any of those?
So what were some of the things that affected your detection probability? What were some of the factors that made it easier or harder? True. Yeah, so where you were. Obstructions. Yeah. What else?
What else affects your detection probability? Any differences between the 4 species?
Raise your hand.
White is hard to see?
White is hard to see, especially when it's on a white background. So a brown butterfly flying around in a swamp is hard to see when it's on a muddy background, maybe. What else?
Or behind the shades.
Or
That's
behind
right,
the
yeah.
shades.
And now it got dark, so you can't really see the shadows anymore. The lighting.
So our hope was that you would start to understand a little bit about how, like, people always think it's really easy to go outside and count animals. And it is like one of the hardest things that we do is trying to actually understand how many of a particular species are somewhere. And all of those things, how big they are, how fast they move, how they contrast with the background, all of that goes into affecting how good we are at detecting them and therefore how good we are at counting them. Yes, there are other butterflies in these areas, and there are at least 3 other species that look a lot like St. Francis Satyrs, but they all fly a little bit differently, so over time you get better at telling them apart, and you don't have to spend as much time like following them around and trying to figure out what they are.
And we use binoculars also to help us. So sometimes they look almost exactly the same, but they have a slightly different shape to their eye spots. And so you sometimes really have to look exactly at those eye spots and count them or look at the shape to tell the difference.
Yeah, one last question. Yeah, so the question is whether we made all this up and we are the only ones who use this, or if it's used more broadly. But this double observer method is used a lot for bird point counts. In fact, the bullseye data sheet that you have is used for birds, where pairs of observers stand at a point for like 5 minutes and listen for all the birds that they hear. We use it for butterflies. Some people will use it for like plant surveys. You have 2 people walk a transect and looking for individual plants, but that is not as common. It's mostly used for birds. But there are other methods like marking individuals and then trying to catch them again that people use for like small mammals. They'll catch them and they'll shave a little pattern on their back so you know you've caught them before. So there's lots of different methods to kind of get at this issue. Cool. Well, I think that that's all we have time for tonight.
Thank you for coming.
I hope you learned something about butterflies.
All right. Thank you, Erica and Elsita. It was really interesting learning about the St. Francis Satyr. Thank you guys all for coming, and thanks to our online audience tonight. We would like to thank the wonderful cafe staff, as always, for giving us free food and letting us use the space. And don't forget to follow Teen Science Cafe and mention us on Twitter, Facebook, and tag us on Instagram. All right, and our next Teen Science Cafe is November 3rd with Dr. Patrick Trueheart here in the cafe, and we're learning about getting serious about the sun. We hope to see you there.
If you love science, check out the Adult Science Cafe on October 12th. It's called Who Needs a Driver, where 3 professors will be discussing the future of self-driving cars. There is also an internship fair on October 22nd where teenagers can meet museum representatives to discuss internship opportunities, among other things. And before you go, there's a survey on your table, so we would love to know how we did today. So if you could please fill it out, that'd be great. And happy Halloween.